The Experiments Proving the Existence of Auxins by KayScience.com. I'm now going to explain how phototropism controlled by auxins in the shoots of plants was proven. So if the shoot is intact, the shoot of a plant is going to bend towards the light. So if the shoot is intact, the shoot bends towards the light. This is because the auxins move down the plant to the shaded region and cause the cell elongation in the shaded region, causing the shoot to bend towards the light. However, in the next experiment, if you place a black opaque cap on the shoot of a plant, the shoot tip is covered with a black cap, and you add a directional light source to it, the shoot will grow straight upwards. It will not bend towards the light. So there is no bending in the shoot of the plant. There is no bending because the auxins, they remain in the shoot tip. This is because this is a shaded region and there is no elongation in either side of the shoot, so it grows upwards without any bending. In the next experiment, a transparent plastic cap was placed on the shoot tip. So now you've got a transparent cap covering the shoot tip. So if you add a directional light source to the shoot, then the shoot is going to grow towards the light. So there will be bending in the shoot. So that means the shoot is going to bend towards the light. This shows that auxins are sensitive to light and will move down to the shaded region of the shoot, causing the cells in the shaded region to grow quicker than the non-shaded region. Darwin determined that something in the shoot determines the direction that the shoot grows. He also determined that the shoot grows towards light. It's question time. Attempt these questions to check your understanding. I'm now going to go through some of the later experiments that proved phototropism. In this experiment here, the tip of the shoot was removed, and when a directional light source was placed next to it, the shoot just grew straight upwards without any bending towards the light. This showed that whatever was controlling the bending of the shoot existed in the tip of the shoot. In the next experiment, the tip of the shoot was removed and replaced with an impermeable block between the tip and the shoot when the shoot's tip was replaced back on. So the auxins that exist in the tip of the shoot could not pass through the impermeable block down the tip of the shoot. So the auxins could not move to the shaded region of the shoot. Therefore, there was no bending. In the last experiment we're going to go through, a permeable block was placed between the tip and the shoot. So the tip was removed and replaced with a permeable block between the tip and the shoot, and then the tip was replaced back onto the shoot itself. Because the block was permeable, the auxins could move down from the tip down the shoot to the shaded region of the shoot. This meant that the auxins could stimulate the growth of cell elongation in the shaded region, causing the shoot to bend towards the light. So the auxins moved to the shaded region of the shoot, and then the shoot bends towards the light. So the auxins moved to the shaded region of the shoot, and then the shoot bends towards the light. So what did experiment 1 show us? That auxins are produced in the tip of the shoot. So what did experiments 2 and 3 show us? Well, they showed us that auxins move through the plant. Also, how auxins affect the direction the shoot bends. It's question time. Attempt these questions to check your understanding. Press pause to practice using those key words. The answers will follow. Press pause to go through your answers and make any corrections to your mistakes. Visit KayScience.com for more free videos, worksheets, and quizzes at KayScience.com. And don't forget to like and subscribe.